to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 9th episode of the Weekly Bioanalysis. My name's John Perkins. I'm the Senior Scientific Advisor for all things related to LC-MS technologies. I've been with KCAS for about 8 months, but I've been in the bioanalytical sphere for close to 25 years, so I have a ton of experience to call on. I'm here with my co-host, Dominic Guarino.
Hello, my name is Dominic Guarino, and I'm the Senior Scientific Advisor for all things related to ligand-binding assays, biomarkers, and flow cytometry. I've been with KCAS for about 7 years. I've been in the bioanalytical space for close to 25 years.
Today's podcast, as always, is brought to you by KCAS, Kansas City Analytical Services. KCAS is a bioanalytical laboratory located in Kansas City, serving the pharmaceutical and biotech industries for over 40 years. Dom and I are senior scientific advisors at KCAS, as we previously mentioned, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS's services. We're thrilled to have you listening again. As mentioned, this will be our 9th week recording the podcast, so if you're interested, the other episodes can be found at kcsbio.com. As a reminder, you can follow us on Twitter at The Weekly Bio and at KCS Bio. As we mentioned before, we kind of started this whole thing because of social distancing getting in the way of normal communication. Even today, Dom and I are in completely different places. I'm in upstate New York, Dom is in Kansas City, and our producers are scattered around the Midwest. The weekly podcast gives a chance to connect with all of you, and we hope it's a chance for you to get to know us, and even more importantly, that it's a chance for you to get to know KCS and the services we provide. As always, a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback from you. As always, we're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. Then we'll wrap up and give you a little teaser for next week, letting you know what is coming in the future. So again, we're thrilled to have you here, and we're looking forward to a fun episode today. To kick us off, Dom is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, so as always, we'll start off our podcast with the news and resources, and it'll focus on some COVID, it'll focus a little bit on some non-COVID, as well as maybe some what's happening investment-wise, and then we'll move on to the main portion of our discussion. Today, it's a focus on the non-bioanalytical groups who help bioanalysis go smoothly. We'll focus on our CSK, or our clinical supplies and kitting. Then we'll take a little bit of feedback and questions from our audience, and then we'll give you a teaser about what's coming up next week. But we're just thrilled to be here with our 9th episode, and got a jam-packed episode, and let's dive right into it. So John, let's move into the news and notes. So we got a jam-packed podcast today. The first news and resources item that we typically start with is the latest on COVID. This week, there was some fascinating new technology to fight COVID, and that's in the Crispr space. And I don't know how much you know about Crisprs, but they're still sort of an evolving and emerging sort of platform. It's coming out of Stanford, and the researchers in interestingly enough, the Department of Energy at the Lawrence Berkeley National Laboratory have looked at a technique called Prophylactic Antiviral Crispr in human cells, or what they've dubbed Pac-Man. Creative, that like sort of like the Sonic back in the day, right? And so this Pac-Man combines a guided RNA with a virus-killing enzyme. That's the Cas13 enzyme. So it redirects the RNA to Cas13 to destroy nucleotide sequences in the SARS or COVID genome, effectively neutralizing it. Very powerful stuff, really good stuff. Actually works quite efficiently to date. It helps delivery via the lungs. I think my only caution with mRNAs, and they've historically had some hurdles, is there's some, side effects from them. But certainly if they can hone some of that in, which there's been major advances in the last few years in the CRISPR technology. So looks quite promising.
Yeah, I don't know much about CRISPR, but it's interesting with the mRNAs, how much they're discussed in the context of COVID with, as a number of vaccines are obviously taking that approach.
Yeah, this is another thing where the advances in science are going to pick up the pace substantially due to this outbreak. So I think it's really, as a scientist with passion for it, I absolutely love that things are kind of being accelerated due to this. I mean, I know that's say that I wish there was a that's not I'm not saying I'm glad there was a pandemic, but it certainly has really accelerated science. And I just want to define what CRISPR is. It's clustered, regular, interspaced, short palindemic repeats, whatever that means, right, John?
I need to do some reading on that. Definitely, yes, yes. I've seen the term and I haven't spent any time looking into it. But yeah, I definitely need to go there.
Just really interesting stuff. And again, I think it's wonderful that we have universities such as Stanford with brilliant people working on these things and just another, ray of light towards a vaccine. So maybe staying along those lines, let's talk a little bit about what the latest on Moderna and Merck are.
Yeah, so interestingly, today, Moderna announced a couple of days ago that they're actually planning to move into phase 3 in July. So their phase 1's still ongoing, their phase 2 is planned, and they're looking to go phase 3 in July. But both Moderna and Merck are now looking ahead, and now their concern is possibly how they actually ramp up production to produce vaccine if the vaccines that they're testing are successful in the clinic. So Moderna are primarily working with Lonza as a supplier because Moderna don't have the capacity to do it all themselves. But they cited a number of things as actually they were worried about in terms of manufacturing space, equipment, raw materials, and people, with specifically raw materials as their concern because the raw materials coming from all over the globe. And then Merck's take was actually they're really worried about if they can find all the people they need because it's the expertise in producing this vaccine scaling up to produce vaccine is also key.
Yeah, and Merck resonated that as well with just, hey, you can pour resources at it, you can set expectations, but if you just simply don't have the manpower to do it, then you're going to hit a roadblock. And that is really what I'm reading here is that there's just going to be, you got so many people running, at full speed with plenty of resources towards a vaccine, but there's just not enough skilled people to make the vaccine and probably not enough skilled people to research the vaccine. And that'll be the real obstacle here. Of course, I still feel like there's some challenges with just how quickly you can bring an effective vaccine into market. An interesting side note in that article about the resources from a professor at the University of Washington indicating maybe there aren't enough communities that are even willing to try vaccines. And that's even another interesting sort of obstacle, meaning there might not be enough people willing to try an exploratory vaccine, although I think time will tell with that one.
Yeah, I'd be surprised. I'm sure there's going to be volunteers to go for this if it means like return to normality and it's a vaccine that will help us get there. I'm sure people will be up for it.
Hey, John, I mean, do you know how long the Gardasil vaccine was on the market before it hit mainstream? I mean, it was available for almost 7-8 years before people finally started trusting it. And now, of course, I think it's a routine vaccine for teenage, both girls and boys. So, but you're right. I think in these circumstances, the mindset has changed.
Yeah, it's like you said about the exceptional circumstances have driven research more. Maybe exceptional circumstances may drive public perception of vaccines more if they can see the benefits to it.
Yeah, I think you're spot on. It was just I think it's an interesting side of things that I had really never even crossed my mind that like maybe people won't take a vaccine. What's the next item on the agenda here for the news and notes, John?
So hopefully this won't be a topic for much longer. But we're back to hydroxychloroquine. But there's been this debate back and forward about its efficiency and whether it's actually doing anything. But the latest information is from a massive trial in the UK, which has actually got a number of arms looking at a number of different treatments. But they did a readout of the they unblinded the hydroxychloroquine arm and did a readout of that and basically showed no difference with hydroxychloroquine and just any typical approach. So the overriding conclusion of the recovery trial, as it's called, is that hydroxychloroquine really doesn't do anything. So hopefully that will, like I say, hopefully that's a non-subject for the future for us, and we can talk about much more interesting topics.
Yeah, I think it does really start to drive a nail into that coffin. I think I wouldn't be surprised if someone tries to counter it. That's just how, the nature of science and publication is. But that study did really convince me that the benefits for hydroxychloroquine are probably nominal to none at all. And let's see if remdesivir has a clinical trial. There wasn't much news about that, although I think a lot of the trials are indicating that it has some positive effect in a subset of patients, but nothing as large as this trial. I'm sure someone's working on it as we speak.
Yes, I think with the hydroxychloroquine we've had now, both one in the UK, randomised blinded trials, both in the UK and as we talked about last week in the US, that have both come to the same conclusion. So, like I say, hopefully that one's done. So I'm moving on to the next topic. So another interesting back to Madagascar again, and this was something my wife found that the education minister in Madagascar has been fired over plans to order more than $2 million worth of lollipops for school children. And the reason why I wanted to do that was in terms of giving the herbal tonic COVID organics that the Madagascan president is promoting as a cure for COVID. It actually has a bitter aftertaste, and so the lollipop was seen as a potential counter to that. But no more to say on that other than that it's really really interesting the way these stories develop as as things move forward.
Well, wasn't the minister fined for the 2 million lollipop order or something, right?
He was fired. He lost his job over it.
Well, as a side note, the Gardasil vaccine for HPV, which I know a lot about, at one point they had a version of it that was edible in like potatoes. It wasn't quite as effective, but this isn't the first one to try to use some sort of I don't know if it was a vaccine, but some sort of treatment that was edible. But what a big gaffe, and something that we can chuckle about. But I hope the people of Madagascar are safe.
Yeah, sadly, I mean, like I mentioned, I go to the Johns Hopkins site, and they I think last couple of weeks we talked about, and they had 2 deaths, and they're now up to 10 deaths. I'm not sure many cases, so yeah, they're not going in the right direction, sadly. So, but it is interesting that other countries have also ordered this tonic from Madagascar, but yeah. I don't think it's really the answer to where we're going.
But who knows? I don't know about the tonic itself. Might have some kind of natural protection to it, but it's probably not long lasting. And certainly a lollipop is outrageous, right?
So the root of it was an anti-malarial. The lollipop is purely to make it more palatable. So I think it's you drink the drink and then you have a lollipop to take away the aftertaste, and that was the solution for the children. But yes.
Oh boy.
Another recurrent topic is masks, and it actually looks like what we're seeing is more studies coming out to say that possibly masks are the single cheap solution that can really help transmission. There's a study done in Germany that they actually looked at, I think, the patterns between public transport before and after people going on with masks. I think that's the basis of it. Yeah, they basically looked at staggered introduction of masks in shops and public transport across Germany, and came to the conclusion that masks helped. And in parallel, also, there was another study that was looking at the basically how COVID is transmitted, and saying that looking at again at data across the US, China, and Italy, concluding that it's mainly airborne. And then both these studies in parallel came to the conclusion that masks really reduce the possibility of transmission by about 40%. So, I mean, it looks like there's more and more information that it really is pointing to masks as a cheap preventative.
In the Midwest, I don't know, Missouri, I know one of our producers is thrilled. Missouri's wide open for business now, and I don't know if there's a lot of masks being worn there, but certainly the masks here in Kansas I'm seeing less and less of them. I think people are starting to use them less and less. I even think the social distancing is starting to kind of fade. That one really troubles me. I'm still. I think the mask has some like efficacy. If I'm on a public transport and I got to take a train ride for 5 or 10 minutes, I'm wearing a mask. I think if going to a grocery store, I bring it. I tend to wear it. But if you're sitting someplace in a confined area for more than 10 minutes, I think a mask becomes. I don't know how helpful it is. There's been a lot of studies around airflows and what it does. It protects for a certain amount of time, and then I've read a lot of the studies that you are referring to, and only time will tell. I can assure you here in the Midwest, the number of masks that are being worn is nowhere near a coastal level. Like, talking to my family in Massachusetts, you cannot go anywhere without a mask. Well, yesterday I had some car issues, and I stopped into a place to grab a cup of coffee, and no one was wearing a mask. I mean, some people coming in had them on, but absolutely no one sitting. And there was even some lack of social distancing in the tables, and that concerned me a little bit. I was fortunate enough to not be very close to anybody. I think it's one of those things where I know there's scientific studies, but I think the reality is we're going to find out real fast here. And just before I move off the topic, I know there's been huge spikes in certain states like Arizona, but I wonder if some of that is just they've got availability of resources to test people. So if you look at how like Kansas progressed, we had a massive spike and then it's leveled off substantially and our death toll is really staying low. But I think that was a testing issue. And so what does a positive response really mean? And there's still some TBDs, but
I think in some states it is definitely increased in testing, but I know that Alabama was cited as again somewhere else I read Alabama has seen increasing cases, but it's definitely not increasing tests. So it's a mix. Some states yes, some states no. But here in New York, it's masks. If in any situation where you can't maintain social distancing. But I had a major milestone this week where I had my first haircut in 3 months. The drill of getting your haircut has completely changed. So basically, I had to wait outside until I was invited into the hairdresser, and then there's screens between each of the people, and everyone's in masks. I was made to sign my name, give a phone number, and other contact details in terms of if anyone in future tests, they can then trace people and let them know what's happened. And then obviously there's disinfectant routines between each of the customers too. So it's very interesting to see how in that situation it's taken very, very seriously. I don't know what a hairdresser's like in your area, but that's how it's been dealt with in New York.
yeah, I'm not sure. My wife shaves my head at home, and I'm not taking that risk to go to a.
And on an interesting note, one of the things, one of the an industry, that has really been badly affected by COVID is actually the music industry. Back in, I think I was supposed to, in May, my wife and I are supposed to meet up with our eldest son to see the House of Love play in Philadelphia. And then obviously as it got closer, we got the note to say that had been cancelled and it had now been rescheduled to next year. But that's true of all bands. I mean, I know it's possible to do like Patreons for musicians and things like that. I got an email from a Welsh band called the Joy Formidable, and they are asked. They basically have a bunch of different packages that if you pay, they can give you access to different specialist content, etc. But also in terms of bands trying to make money, it's like can they leverage this? So I got a link to a King Crimson website, and this was a few weeks ago, that if you bought more than a certain amount of vinyl, CDs, whatever, or merchandise, they would give you a free in the coat of the Crimson King face mask, so it's the very famous album cover where it's got what they call the schizoid man, who's sort of basically yelling on the front cover, and the mask is basically the yelling mouth, and the person promoting it on the website was the bassist of King Crimson at the moment. But then not all bands take this same approach. Jethro Tull, who are very sort of controlling of all their merchandising, etc., posted on Facebook that they were really not happy with third party vendors selling face masks using Jethro Tull sort of imaging, and discouraged people from using it, particularly as they weren't as effective as like 3-ply masks, etc., etc. And so they maybe they should read some of these latest studies to say that even if they're not 3-ply, that there's still some effect to their masks.
Well, I guess you won't see anybody sitting on a park bench with a mask on. Not that I think we're dating ourselves with some of this music, too, John.
And I wouldn't be pushing for that either. It's like, yes, absolutely. Yeah, once you're out in the open, I've got no concern about it. Certainly, we walk around the village with no issues, and I do get the tall reference.
Yeah, no, I figured that. I don't know the age of our audience, but in prison, I vaguely remember them and Jethro Tull similarly. But maybe we'll move on from the music side of things, and or even the COVID. Let's jump into some non-COVID related stuff. And this is exciting for me in terms of Gilead has an announcement around one of their small molecule drugs, a JAK1 inhibitor for active psoriatic arthritis. Excuse me. And the recruitment has resumed. So this is a great sign to see non-COVID clinical trials starting up, right? So I think this is fantastic. I hope we see more of it. But it was good to read that and refreshing to see that we might be, again, sort of coming. Look, I think we should focus on COVID, obviously, but I think we can't just let go of all the drugs, all the other, disease states out there. And psoriatic arthritis is one, but in particular, as we touched on last week, it's really those rare rare diseases and orphan drugs that are taking a bigger hit. But this was encouraging.
Yeah, I think it's critical that really research goes ahead on as much as possible. We can't let COVID derail things. And I think you mentioned the rare disease. There was a really excellent article in the Washington Post earlier this week, which I would encourage anyone to search out, about the effect of COVID-19 research on rare diseases. They are being impacted. And the big deal here is these are conditions that only affect small pockets of the population, but they are, so many of them are degenerative, where people's quality of life can rapidly decline. So any time lost in looking for a cure, it has a massive impact on people's lives. So it's almost like the one pocket of research you don't want held up, but sadly that's been the case, because like I say, it takes a short time and people can drastically change from where they were. So hopefully, the focus can get back on rare diseases and get them moving forward as they need to do.
Yeah, it's something that I've had the pleasure of helping in the rare disease space. It's one of the most rewarding things to see some children that have like some debilitating lack of enzymes, and their motor skills go completely, they're gone. They go from like being adolescent children that can run around and pick up a pencil, and suddenly over like a 3 to 6 month span, they're because of this lack of an enzyme, they suddenly can't do any of that, and then they get an enzyme replacement, and now they're kind of returning to normalcy. And I've gotten some handwritten letters from individuals that were impacted on drugs that I've worked on, and it is one of the most rewarding things that you can do as a scientist.
I understood. It is an absolutely fascinating space when you end up talking to people about rare disease work they're going on. Because for someone, most of us are not aware of any of these until we actually get to talk to someone who's working on it, and it really is tremendous what the change it can make to people if you find the right therapy.
Yeah, absolutely. John, why don't we jump into some recent investments? I thought it was interesting. So, well, not recent, but some stats came out around in 2019. The top 10 pharmaceutical R&D spenders was a whopping 82 billion in their research for new drugs, diagnostics, and vaccines, and that was up by about 4 billion from the previous year. 2019 was another really big year for FDA approvals, right? There were 45, is what this article said, but the record of 59 in 2018 But the record of 2018 was the record with 59. The 45 in 19 was the third biggest in the new molecular entities, the NMEs. So interesting. I thought that was good to see, and I'm sure 2020 will take a pretty big hit with what's happening now.
But no, well, at least at the moment we're on pace to match 2019. I know we touched on that last week. But again, it's that's on the basis of everything going at full speed, and we have seen obviously that we know that there's a number of trials that have been held up. So hopefully it doesn't knock things too much.
Yeah, you're right. Maybe it's not 2020 and maybe it's 2021, but then again, who knows? Maybe due to some of the positive light in the pharmaceutical industry, maybe we don't see a blip. I certainly hope we don't, but my sense is the 3-month hiatus here will eventually catch up to the curve. And recently, I don't know, as a side note, I heard that a company in Boston finally, I don't know exactly what's happening on the coast in all companies, but one of them opened up this week, and that was encouraging. They were allowed to come back into the laboratory using social distancing for the first time in, like, I think it's almost 10 or 12 weeks, sort of how we've been doing things here at KCAS, and that was encouraging. So let's hope that in the age of virtual communications, let's hope we don't, being able to work at home, we can certainly regain our traction despite maybe not having as many people in the lab.
Yeah, it would be nice to be out there again. Another big piece of news, we haven't discussed this actually, but it was I think it was a couple of days ago, AbbVie and Genmab, who are a Danish company, announced that they'd entered into a broad collaboration to develop and commercialise three of Genmad's next generation bispecific antibody products. It's a huge deal. I think it's 750 million up front, and then milestones up to over 3 billion. So, and actually, looking at it the following day, the stock market reacted quite positively to this deal. So it looks like a good move for AbbVie.
Yeah, I did read about that. That was encouraging. Bispecifics are very powerful drugs. They're very challenging in the bioanalytical space. In fact, we've got a nice publication from the AAPS PK focus group that I sit on trying to go over some of those challenges. Maybe let's round out. I think there's one thing that made me laugh in terms of our last news item. So there was a big stir in the industry around a potential AstraZeneca wanted to merge with Gilead. So they came up with the name Gilead, which was funny. Gilead. Anyhow, yeah. So Gilead is worth 96 billion, and AstraZeneca is 140 billion. So it would have been the biggest deal of all time. The largest deal was actually BMS's $74 billion acquisition of Celgene. But the very next day, AstraZeneca came out and said they ruled that out, this prospective deal, because it would distract from the company's pipeline of ongoing COVID vaccines. And that was a British report. But this is how fast the things can change. One day it's like Gilead and AstraZeneca is going to acquire Gilead, and then the very next day it's like, nope, that's not happening.
That was a bizarre cycle, definitely. Yeah, they up and down so quickly. Yeah, that odd one, that one.
I just what it did is I didn't realize the value of AstraZeneca and Gilead being so high, and then also I found it interesting that BMS's acquisition of Celgene for 74 billion, and that was a big, obviously the largest ever, and still has really helped BMS in the immuno-oncology space.
Yeah, it's always interesting to watch, the large pharma, particularly as they move around, change the research emphasis and the effect it has on their pipeline. I mean, I think a few years ago, AstraZeneca's pipeline really wasn't doing well. They had a number of hits where things got to late phase but died. But it looks right now that they're on a roll and doing pretty well.
Well, that'll do it for the news, John. We got a really exciting main portion of our podcast. Week 9 of the podcast. It's an exciting topic. It's one that, having been at KCS for a number of years, I certainly knew quite a bit about our CSK. I think you were drinking from a fire hose, realizing just how much we do. But maybe we can jump right in. What do you think? You ready for it, John?
Yeah, so basically, I'm going to probably let you get going, but what we wanted to talk about is we focus a lot on bioanalysis, but the bioanalytical teams can only really function with all the groups around them who are part of the process. So there are other groups. We'll mainly talk about CSK and we'll go into that more. But there's other groups such as our sample login team, our test materials management team, our metrology team. We have QC, QA. All these groups are get hands on the product. IT, of course, is essential to what we're doing to really help move us through. That if without any of these groups, we would fall apart in the bioanalytical part. But like I say, we thought we particularly focused to start with on our clinical sample kitting group. The reason for focusing on them, although they have less impact on on our bioanalysis unless they're involved in the clinical sample kitting part. But actually, when we actually started hearing about COVID trials, etc., as the obviously the pharma industry started to look at how they could address this virus, they were the first of our teams to actually be involved in supporting COVID work. So they have to date have supported a couple of clients and multiple studies, and it's the their role is to sorry, I'll say that again. Unless you want to jump in at any point, Dom. So, so, yeah, that's fine. So their brief is basically providing clinical study kits or study kits to support clinical trials, and these will be tubes to support pharmacokinetic samples, PD and biomarker samples, and it's not just fluids. They'll produce kits so you got sampling of tissues if they're relevant as part of the clinical study too.
Yeah, maybe I'll even take us a further step back in how we got to being a clinical kitting supply group. And it started way back from inception. KCS, a 40-year company, right as PK analysis became more and more regulated for small molecule, we started to solve a lot of problems with stability of metabolites. And so we got creative with adding stabilizers to certain tubes so you could draw right into them. And so then, how that created our clinical supplies group. So now we started taking tubes and supplying them with certain stabilizers and then sending them to sites. So that's how we started doing this. It actually helps flash forward, that's probably 20 years ago now. If you flash forward to today, it's become a real important part of helping with efficiencies because now we're providing barcoding. We're using our Watson LIMS system. It actually has some efficiencies with not just PK, but into the PD and biomarker space where we're providing very specialized tubes. One is something called a CPT tube, and that's used for quick isolation of peripheral blood mononuclear cells. So the site could just spin it and ship it at room temperature. They don't have to go through the rather laborious job of having to harvest it, putting in freezing media and putting it at liquid nitrogen or minus 80. A lot of these sites don't have it. So that solves with helping to preserve your peripheral blood mononuclear cells for pharmacodynamic assays and biomarker testing. Additionally, we supply something called a PAX gene, which is for mRNA. We have a cell-free DNA tube for preservation of DNA. So it's really helped round out and having knowledge of this, and we have the ability to have those tubes shipped back to us, harvest them, and store them for you. It really helps with sample logistics, sample preservation, and all of the things that's really the fuel to bioanalytics. If you don't get the sample into its right storage as quickly as possible, then you're going to fail on all levels. It doesn't matter how good you are bioanalytically if you don't get that first step going.
And again, it does really help to have the samples go out with that Watson key, then comes back in, and we're using the same system rather than when you have samples come through a different central lab where they can be using a different system, and it just the the logistical flow isn't as easy at all. So it definitely smooths out the bioanalysis if you're doing the kitting up front.
Yeah, and I mean, absolutely. This was a service that was growing, I think, over 100% prior to COVID, and now it's just exploded. Like you said, to date, we've two clients, three trials. As a little bit of a side note, we've got our first biomarker data from one of them, and it's not surprising. These inflammatory cytokines are off the charts in these patients, and most notably, one of the small molecules has really dampened that. Lowered it substantially in the 4 patient sets that I've seen. And the great part about this is it's a collaborative thing. So hopefully we'll have the data readily available and we can share it with the public, showing how things like interferon gamma and TNF alpha, all these inflammatory cytokines, drugs are helping to treat it. And this, these are like, unlike some of our other drugs that are on the market, these are things that might be able to help. But back to our kitting and supplies team, I think John, you touched on how it can help with workflows. I talked about the number of tubes. And then we've been doing it so long, we're very good at the manuals. We can go to sites and show, kickoff meetings where Trish Landman runs the group. She's been with us for 25 years or maybe longer. I think it's around 25. And she can go and give a presentation in front of, say it's a multi-site, 20, 30 sites. She'll give a presentation on the manual, has huge successes doing that. She's also, understands a lot of the challenges and obstacles and has really gotten creative with, as the number of biomarkers increases, the number of specialized pharmacodynamic tubes, she's come up with creative ways to ensure that sites can handle, they're used to maybe just seeing a PK tube and stuff, or maybe even PK and ADA in the biologic space. And now you got 3 specialized tubes. Well, she's gotten really creative with stuff like color coding schemes as well as just the manual itself. So we just got a lot of depth there, a lot of expertise, and
that is key, the color coding, because for us with clinical trials, the success of our ability to do bioanalysis depends on the clinics being able to take the samples properly and deliver them to us for analysis. Everyone recognizes that people in clinics aren't the experts in terms of preparing samples for downstream bioanalysis. So if you're kitting and up front is really clear in terms of instructions for the clinic and how they take samples, then you're going to minimize errors. So you're then ensuring quality downstream, because the clinic has been able to do their part of the process smoothly.
Yeah, and I'll even add, we have been doing it so long that there's not a matrix or tissue type we have not provided a kit for. I know we've provided needles for CSF. We've gotten creative ways to how to aliquot that CSF sterilely, because a lot of times that's done like right in the surgical room when they're doing it. I mean, we have, and then when it comes to tissues, again, we're very experienced with all sorts of, whether it's a paraffin embedded block or a flash frozen block, or however it might be. We have recommendations on certain medias in case you want to drop it in that. I mean, it's really a powerful service that we can help with ensuring that, again, as I mentioned earlier, if that component isn't done right, it's like the fuel. The engine is not going to run bioanalytically if that's not collected right.
Yeah, and it's interesting that the service of treating tubes with stabilizers, etc., is still something we do, but that's where CSK becomes more integrated with the other operation, because our lab staff are the qualified people for preparing those treated tubes. So it isn't a standalone service. It's very much working with the other teams, and it's like we've said on other podcasts. We see the benefit of us all being under one roof because we're this collaborative team. And here's the intersection of CSK and their operations team.
Yeah, and even this morning I had a call before this podcast. We looked at someone had some urine samples that they worried about that were coming from Australia clinical trial, and like what that they were looking at us. And as I mentioned, we had clinical supplies because I talked about tube types and all this sort of stuff that they're just sometimes unaware of, like some of the challenge. They know they got a challenge, they just don't know what all the let's call them obstacles are, but we have solutions to them. And really it starts with, doing a feasibility study, John, and that leads into us recapitulating what the lab can do, the clinical site does, so that we can ensure they get to us. And that's a real small investment that, and again, in this case, it was urine. It really is like powerful stuff. And you could even get a sense of relief sometimes when you're like, oh, I didn't know we could do that, so it's wonderful. And as you mentioned, having it all under one roof really helps with that.
And again, that plays into another piece. Obviously there's a lot of time and experience has gone into this, and it's that recognition that not all analytes need the same treatment. So when it comes to PK kits, yeah, we can put all the samples for a patient in one box that's shipped out all together. But when it comes to some of the PD markers, where you've got much more limited time in terms of stability, etc., we can go down to a daily shipment routine to send out tubes when necessary for that next sampling. So it's not at all a sampling up front, because again, it's more not thinking about making it easy on us. It's what makes things easy for the clinic, because really ultimately it comes down to, if the clinic can deal with it successfully, then we'll all be successful.
Yeah, that's right on, John. I think that's such a well said. Um, Continuing with the kitting, one thing that we've got a lot of experience with is shipping internationally. I think this can be quite difficult, meaning we work with a third-party CRO in other countries, and we help them manage the logistics on their end. And so, on a side note, a funny story. We were doing some work in South America, and I won't say specifically what South American country, but the supplies kept getting stolen. So, Trish, like we send 10 sets of patient kitting for 10 collection unit, and we only get 3 of them back, and yet they're asking for a resupply. So literally they were taking the tubes and the needles. And so Trish had to help manage, the team had to help manage that. And really another, you don't think about maybe poaching of supplies, but, and I don't, maybe it wasn't malicious. I think sometimes it just happens, but that's another added benefit of, having the lab that's doing the bioanalytical component providing the kitting.
Mm-hmm. Yeah, and the other thing we know from international is, again, we're trying to avoid COVID, but we can't avoid going back to it. It's made things substantially more difficult. So the third party CROs are really useful in that case, and even more useful is World Courier, who we anyone who's been in this business for a while knows that with international shipping, if you want a quality partner is World Courier, who will refreeze samples as necessary. They've always been a good partner to work with, and so we're working with them in terms of figuring out when we can ship to countries who are currently closed.
Yeah, very true. We've been getting them from Italy. We've gotten 2 shipments pretty seamlessly within a few days. Very happy about that, and that's the biomarkers that I was alluding to earlier. Um, what else? One of the biggest issues is protocols, right? Um, Not being completed or missing certain pertinent details, meaning and that relates to, if you have for us, if we're doing the lab manual and the bioanalytical, we really do merge the two, meaning Trish and her team will sit in on our technical calls. They'll get an understanding of any feasibility studies we've done. They'll listen to the client. They'll listen to the scientist. There's interpretation of that, and then they build the manual off of that. So that really helps with increasing the success, right? It's all about percentages of success is how I think of it. And if you allow us to do more and more for you, I think it really increases the potential for success. And I don't just mean us for it. It's an investment, right? So as I was alluding to, when we do feasibility studies to help the clinic understand how to collect for PK, ADA, biomarkers, and pharmacodynamics, we can avoid any sort of things like, hey, there's a lot of times we've done certain studies for cytokines where it said, hey, we can't store that this way, but here's an alternative way. And again, if they didn't make that investment, they would have, at the end of the study, been looking at data and said, oh, wow, well, this doesn't look very good. So just simple stuff like that I say simple, but that sort of investment is, I just don't understand why you wouldn't want to make that for your study.
Absolutely. Yeah, the other part is, it's all about down to communication and all this, and then flexibility is another big part. And I know that, we we have constant discussion and collaboration with the clients, but things change in terms of study design. We know that your protocol is fairly fluid until it's finalized, and we've had instances where we've had to turn on a dime. We've actually got kits complete, but there's been a last minute change. Yet we've still managed to get kits out on time to meet customer deadlines. So it's all part of that consistent approach. We collaborate, we're flexible. That's our model where we're focused on client service. So CSK is consistent with bioanalytical in terms of our approach to this.
Yeah, and absolutely. I mean, with the way COVID is moving so quickly, you just described a scenario that happened this week. In fact, I think I just saw a manual go out from Trish that said, based on a call we had Monday, they adjusted their protocol and kits, the manual's ready to go, and she'll be shipping kits next week. And that's just an incredible amount of speed and flexibility to be able to, within days, pivot from where you are.
So I think, is there much more to say about CSK?
Yeah, go ahead. I think we're ready to move on to just maybe some other departments. I know, I think we've covered CSK. It's a wonderful group, but all the groups are great. Especially, let's start with the test materials management team, John. I mean, they do more than just bringing in the materials and reagents. They're now, working on the accessioning piece, which frees up our scientists. And what I mean by that is, they'll take now the samples, they'll count them, they'll do a visual, make sure nothing's broken, and then the accessioning component and entering it into Watts and Limbs, and they'll help with some discrepancies so that work, the onus of doing that comes off of our scientists and analysts. And so the scientists and analysts, all they're going to see is they provide a plate map into Watts and Limbs with the samples that they want to run, and then test materials management is making it happen for them, saving a lot of time, and that's actually helping to drive our making us more efficient in terms of our pricing. And it's really a very powerful group in the TMM side of things. Before we move on to other groups, you want to share anything about our test materials management team?
I honestly haven't had much interaction with them, but I do know from previous experience, if that group is functioning right, then the whole place is functioning right, because everything is founded on the group who are responsible for samples and materials and help and that sort of check in and getting everything in place for analysis to proceed.
Yeah, and it's so true. Just it's something pull through wise. It's all the way from the analysts getting the samples to enter, into QAing, because that starts it with how they're getting into Watts and Limbs, all the way through accounting is dependent upon TMM, because that's ultimately how we invoice them. So it is a critical step.
And not even just getting into analysis, but the being on top of the samples and knowing the storage times. It's critical for getting samples out the door for once studies are finished, so we're not all backed up in terms of materials, so we can keep that flow through the facility.
Very true. The other is metrology. There's several on the list, but let's move to metrology and touch on that real quickly. And that's our, they're our instrument calibration team. And again, that group takes a lot of onus off of the scientists and analysts, because, we're in a GLP environment and every instrument has to be logged, monitored substantially in terms of daily, weekly, and monthly routines. They're a great group. They have a lot of talent. They have the ability to even, we have service on the instruments, but they can, in particular, Lahal, who's in that group, has a lot of engineering background and is able to even, sort of fix some of the instrumentation. So really a very powerful group, and one that really does help, again, that ultimately what we're about is delivering data.
Yeah, I mean it's key, and this all comes down to, like, if you I mean, I started 25 years ago, and when we actually started to use an instrument, there was virtually no qualification. It was the engineer came in, installed it, showed it was working, and off we went. But there's been much more emphasis on the IQ, OQ, PQ of instrumentation before you can even consider it for GLP work. And so this group is absolutely key. So we although we're focused on new technologies, etc., but that new technology comes in through the door, we can't immediately start using it. We have to make sure that that instrument is capable of doing what it's described as being able to do, and that's what the metrology group helps out with. So again, critical, absolutely critical to us as bioanalysts, and not putting the onus on the people, the bioanalytical people who are much, at least in my realm, much more focused on extraction and LCMS. So we're hugely dependent on that team.
So, I had the good fortune of starting in 1994 at a biotech company, and we kind of grew up in opposite worlds. It was we were treating autologous, uh, we were treating metastatic renal cell carcinoma patients with autologous T cells, and I worked in a class 10,000 clean room, and every piece of equipment had calibration to it. The difference was it was monitored. It was done by our QA department, which was an interesting sort of component to it. And I'll never forget the day we had a problem with one of our cell counters. It was an old Beckman Coulter impedance cell counter. And this guy came in and we couldn't fix it. We were flushing it and he pulled off the aperture and he blew it out with his mouth. And we were like, oh my God. He's like, what? It kind of hurt you. And it was really even now as I sit here 26 years later, it was a memorable moment. I forget the name of the individual, but him mouth pipetting, of course, that was it. That was a no-no. You weren't supposed to put your mouth on anything by the mid 90s. But really, just a side note that we kind of grew up in different worlds. Being in that was a GMP, GLP at the time.
Sure, I think it was a GLP LCMS back in '94 was really finding its feet, and I mean it found it pretty rapidly. But there's things like I say, instrument qualification was much more our standards and QCs pass on a daily basis. Things are working right. It wasn't so much of the showing that a pump flows at the rate it should and the mass spec does what it should. It's much more comprehensive and has been for a long time. I just like I say, 25 years ago it wasn't there, but it's been in this position for a while now.
I even remember it was actually the irony of this. It was a Streck Laboratories, which ended up working for in the early 2000s. Control that we put on there, 3 levels of control to make sure that that impedance cell counter worked. So come full circle in my life there. So let's move on to a couple other critical departments. One is our QC department. I think that speaks for itself, right? This has really driven down our um, deviations in QA findings and all sorts of stuff because it's really just a standalone department that so the way it works with a batch, doesn't matter what group it's in, it gets reviewed by the analyst or scientist, it gets a peer review QC, it gets then it goes into the QC department and gets reviewed, then it gets the principal investigator's review, and then it goes into QA. Huge investment, moves very quickly, but that QC department that we have is QA trained. And I'm pretty sure Tracy, our director, can show you how we added this, I think almost 9, 10 months ago, the standalone QC department. And it's really improved our the days that it takes to get something through QA as well as the number of findings.
Yeah, that was a model I was used to, and it really does make a difference to have an independent QC group. Certainly, going back several years, we used to do peer review, so it was a scientist reviewing the data, and the key here is it's all about giving customers the reassurance that their data is solid. So we would say it was peer reviewed, which most customers accepted. But to say you got an independent QC group doing this really does help. Particularly as if we're looking at dose escalation studies, you're not at the end. You're sending out data on an interim basis. You're not at the end of the study where you're doing that full review of everything. So to have the data, say for a single dose escalation, goes to QC group, which then goes to the client, that really gives them like say, reassurance that their data is solid, that they can then go and start modelling, maybe to go to the next dose in that particular study. So, a really good thing to have implemented.
Well, the last team that I want to touch on is our validation team, and that's been with us all the time. And they're actually responsible for bringing on instrumentation, and that again, and maintaining it. They're sort of an add-on to the metrology group, but it's a standalone validation teams. We've got so much instrumentation coming in and out that again, it allows for the scientists to focus on the actual data they're producing. And that team, writes all the SOPs. When we just expanded our cell culture suite, added 2 flow cytometers, they were responsible to help drive in that so that our scientists could continue to work without having any perturbations of their workflows. And so that team plays a critical part as we continue to talk about bringing on high res mass spec or swapping out some of our 4000s for 6500s. That's all managed by a standalone team, and really a powerful addition and a nice investment that certainly at KCS we've had that team since I've joined. Discussion today, John. Really enjoyed the topic of, and I don't know of a better word than our support services, because they do so much more than just support. I feel like they're as critical as any sort of scientists and analysts in terms of their day-to-day functions to ensure that we can provide reliable and defendable data. Really enjoyed talking about it. And any last thoughts before we move on to our feedback and questions?
Yeah, I hate the term support services because, like I say, they're so integral to what we do. I mean, at some point, it would be good to have possibly some people on this podcast as guests to talk about some of these different things and have a more in-depth discussion than we're able to provide.
No, I think that's a great idea. I'm sure Trish would love to come on and talk to us. And somebody who's been with the company for so long, she brings a wealth of information, and just she is such a client interfacing part of our organization. She is on the phone daily with sites, helping to remedy things, and being, our team does a lot of project management because of all the issues that come up from sites. And even as I described, to some degree they're like police. She now knows that they monitor, they have an incredible amount of tracking to ensure that, like, if a site's calling for another 10 kits, they better have sent back 9, right, or 8. So even that added sort of feature from the experience that we have. So that'll do it for the main portion. Let's move on to our feedback and questions. John, you want to get us started with the first one?
I would say maybe it's better you get started with the first one.
The first feedback we got was from Terry. Terry says, all I can say is thank you to the KCS team for the episode on hybrid versus LBA. It got our team thinking and asking a lot of questions about how we might move forward with our projects. Don and Frank were wonderful first guests. Yeah, thanks, Terry. It was very enjoyable to have some guests. It was a little more relaxing, that episode, to have someone else doing some heavy lifting. But certainly you can see the depth we have with Don and Frank. There are others behind them in terms of their knowledge for both hybrid and versus LBA, but it was a pleasure. And how about you, John? What did you think of that episode?
Yeah, I always love listening to Don talk. So even though I've been associated with LCMS for so long, and the hybrid LCMS more recently, Don's knowledge is fantastic. So I always like being in calls and stuff with us. So to be part of that discussion was great. Actually, I have to say on top of that, I'd really like to know what the projects are that Terry's wondering how to move forward, because it's always interesting to know what the scientific questions are that are being asked.
Yeah, Terry, send us an email, send us a text, whatever you want. Let us know. We'd love to chat.
So next up, Daryl asks, perhaps improper to ask, so I apologise, but John, I'd like to ask you where you are from originally. My background's a little bit not as straightforward as it might be. So I grew up in Glasgow, which explains the accent, but there's a bit of background to that in that both my parents were from England. So my dad was from Herefordshire, which is on the Welsh border. My mum was from London, and my dad ended up teaching at the University of Strathclyde in Glasgow. I was actually born in Dumfries, just over the border into Scotland. But when I went to school in Glasgow, I would have had what was a fairly straightforward English accent, because my primary influence for language was my mother, who was a stay-at-home mother at the time. Here's a tip: don't be an English kid going to school in Glasgow, because guaranteed it's like having a target on your back, because you will be picked on big time. So I, at the age of whatever, acquired this accent because it was effectively camouflage to fit in with the kids around me, and it's stuck with me ever since.
That's a great story, John. You're just making me laugh with that, John. I don't know. I think every kid goes through some picking on, but that's you did a good job of evolving to fit in. But that's pretty funny.
That's the total truth of the matter. That honestly, that's how it is. I have two sisters, and we all have three completely different accents. But so theirs are much less Glaswegian than mine is. But yeah, this is where it comes from.
Yeah, I don't have anything as fascinating. I mean, I actually I thought I was fascinating in that I was born in Okinawa, Japan. My parents are both from the north end of Boston, but your story beats that with the stick. I mean, I guess I grew up pretty seamlessly in the suburbs of Massachusetts, and didn't have a whole lot of picking on, but certainly had my fair share of run-ins, if you know what I mean.
How long are you in Japan?
5 years. So I was born there in my first 5 years, and then my dad served a full 20 years in the Air Force, and his last 8 years were in Okinawa, Japan, and my brothers spent 8 years of their life there. My sister was born there as well. She was 3 when we left. My dad had a house in Massachusetts throughout that, and we moved back to Burlington, Massachusetts, which is where I'm originally from. And then when he retired, we settled in Burlington. In fact, my brother still lives there. My mom and sister now are in Norwood, Mass. I'll just round it out. And then I got one brother. I got another brother in Colorado. He's outside of Cripple Creek, Colorado. But
So do you have any memory of it at all?
a little, that's funny. I remember the barracks that we were in. I remember things called monkey lizards, a lot of ordnances. I used to find live ordnances because that island got pummeled. So I remember like coming home with like a hand grenade and being like, look at this. And my mom freaking out. I remember bugs. I remember a lot of bugs. I know a little bit of some like Ohio Gazamis, a little bit of Japanese, and remember my brothers being able to sing "You're Happy and You Know It" in Japanese. And then I'll round out with there's a swing set that sticks out that I used to play with a couple kids, as well as some of the weather, of course, was beautiful, and then sirens. So periodically, sirens would go off because of the ordinances that were found on the live ordinances that were found on the island 20 years later. Yeah.
How long are you in Japan? Oh wow, yeah. Yeah, yeah. I was lucky enough that I actually got to go to Japan a couple of times over the last few years, and yeah, it's a great place. Really, mainly Tokyo, but it's still fascinating.
Well, the place we got to get back to is Barcelona.
Absolutely, yeah.
Spain.
There is an update on EBF. They push back date for the deadline for abstracts for oral presentations, etc., and we'll have news in the next couple of weeks about what format the conference is likely to take. Praying it's a physical one rather than a virtual one, but we'll see.
Yeah, and then I don't know if you saw in AAPS, they're making it both. Right now, AAPS in New Orleans, you can attend. It's also going to be virtual. I think that might be a sense of what's happening. Slated to go to what's called the Immunogenicity Summit in October. That should be live. I hope it will be, but you never know. We touched on the volatility of how things are, and only time will tell. But I think over the next few months, we'll continue to get clarity on things. So, John, any weekend plans? I know it's a little we tripped things up a little bit this week with some car issues, so we're having our podcast on Friday instead of a thirsty Thursday. Anything going on this weekend?
So my biggest plan this weekend is not car trouble, but lawnmower trouble. I actually ordered a small hand pushed or hand propelled lawnmower, electric one, except the charger doesn't work. So yeah, I need to get that resolved. So I'll be taking that back to see if I can get a replacement battery pack or charger. I'm not sure which is malfunctioning, but something that's not working. And then the other part, with it being this time of year, my grey VW is currently green with pollen, so that's gonna go. So yeah, it's no big plans. You can tell it's all small stuff. And then actually, my youngest son, who is the one of my 3 kids at home at the moment, is having some friends round tonight, so we're going to practice social distancing with a few beers. So we'll see how that goes.
Oh yeah, that's a fun one. It's really enjoyable to get out and be with some people. In fact, Sunday night I hung out with Frank Spriggs. We saw each other for the first time in 10 months. It was doing his outdoor patio. Had a couple spritzers and had a good time seeing Frank for the first time in a long time. For me this weekend, I got to mention my favorite thing. I have been searching high and low for yeast. I should have picked. Frank meant to bring some. He didn't, but I have not been able to find yeast. We're up to like 3 grocery stores now, and I can't find any.
You should let me know what brand you want, and I'll see if I can find it here.
Folsham's or whatever you have, anything you can get. No, I'm sure I'll find some, but I did make a big pasta sauce. I canned about 8 quarts of it. So I got some pasta sauce ready to go. I just don't have any dough to make. So it won't be a Pizza Friday, which saddens me a little bit. I'm not sure. We might have to make some steaks instead, but got some pasta sauce. I did make a good meat sauce. Man, I'm getting myself hungry. So anyhow, we can't go on a podcast without mentioning pizza, John. It's just impossible.
I barely can let many go past without mentioning Marmite. So actually, news article in the BBC this week that Marmite lovers who haven't been able to buy larger jars of the spread during lockdown can blame the coronavirus. Basically, there's a again, it comes down to yeast, but there's a shortage of brewers' yeast. So there's a Marmite shortage in the UK, and people are panicking. So they only produce 250 gram jars at the moment, and there's a lot of not very happy Marmite eaters. So we'll see where that goes.
Man, I'm so happy to learn I'm not the only one struggling with the yeast thing going on. I wonder, that's amazing that you must have laughed pretty hard when you're like, dang it, Dom's having a problem. I might not get my favorite food.
Well, that was it. I was like, there's no way I'm letting them know up front that I'm gonna bring this up. But yeah, this definitely has to be included.
Oh, let's end it with that, John. Thank you all for listening this week. It's been our pleasure to bring you our 9th podcast. Very excited to go into week number 10, which I think is like a big number, right? It seems like we're going to be bona fide if we can get to 10 podcasts.
We'll be teenage soon.
Yeah. Next week is really an exciting topic. It's where does bioanalytical fit in the drug development process? I think it's a big topic. It's one that I hope we can keep to under an hour. I think you've got 2 guys that have a lot of passion for drug development and specifically how bioanalytical fits in it.
Absolutely. Yeah, looking forward to that. So yeah, I don't think there's much more to say, but apart from, again, thank you very much for listening in, and we look forward to doing this again next week.